Oh, I'll be brave if you're brave. I'll be brave, but only if you're brave. And it could be just you and me. We'll be family. Just wait and see.
Good evening, everyone. Welcome to tonight's Lung Cancer Living Room. We're so happy to have all of you here today. Happy July. Welcome to those of you who are able to join us. I'm Danielle Hicks, Chief Patient Officer here at GoTo Foundation for Lung Cancer. For those of you who aren't familiar with our Living Room program, it's a support and education series created specifically with patients and caregivers in mind, with the goal of bringing to you live and in real time educational talks from key opinion leaders in the lung cancer community, and in a format and a language that's easy for you to understand. We know that people watching tonight come from around the globe and consist of patients living with varying types of lung cancer and are undergoing various types of care. We want you to know that we're here for you and that we believe wholeheartedly that educated and empowered patients do better. So thank you for joining us. For those of you who may be familiar with the Living Room, you might notice that Bonnie and I have different backgrounds behind us tonight. We are on a family vacation, but nothing is more important to us than this community. So welcome from Tahoe. We're happy to have you. For those of you who have also seen who our speaker is tonight and what we're talking about, it's Dr. Pasi, who is MD, PhD, Professor of Medicine at Dana-Farber Cancer Institute. Tonight we'll be discussing updates from the American Society of Clinical Oncology conference that occurs every year in late May, early June, where data is presented on the emerging science. We'll go over information specific to lung cancer in multiple categories, including targeted therapies, biomarker testing, immunotherapies, early stage diagnosis, and small cell lung cancer. So welcome everyone, and welcome Dr. Jänne. Before we get started, I always like to have our speaker give us a little bit of background on who he or she is. I could read bios and go through this laundry list, but I think it's more personal and creates a better sort of connection with the speaker and with the topic. So Dr. Jänne, can you give us a little bit of background on who you are and why lung cancer is your main area of focus?
Thanks, and welcome everybody. And it's a real pleasure to be here today. I'm coming from my home office here in Boston, Massachusetts. So as Danielle mentioned, I'm Pasi from Dana-Farber Cancer Institute. I'm a medical oncologist and a translational researcher. I do both lung cancer laboratory-based research as well as clinical care and clinical trial work, and have spent my entire career at Dana-Farber doing that. And when I started in lung cancer research over 20 years ago, the picture was quite different. We didn't have the therapies or the understanding of the biology of the disease that we do today. And I was interested in it as a disease and interested in also in trying to figure out what can we do for this very common disease that impacts so many individuals, and got started in my career in lung cancer, again, both doing laboratory work and clinical trial work and clinical care, and still continue to do it every day. So it's a real pleasure to be here today, and I look forward to the discussion about ASCO 2021.
That's great. Thank you. And I know Bonnie doesn't need any introduction. And for those of you who are new and may not know, she also happens to be my mom. So lung cancer entered my life in late 2003 when she was diagnosed with stage 3B non-small cell adenocarcinoma. And I know she might have a little bit of something to say about what was or was not available some 17 years ago. So I want to give you an idea, say a few words.
Well, the few words are there was nothing available but chemo and radiation. That was it. And actually, at that time, though, the EGFR mutation was in clinical trial. But I didn't even get tested because it was so new. That was the absolute first mutation that anyone looked at. And I'm not going to waste any more time talking about myself. I just want Pasi to jump in there and start letting all these people all the good news.
That's great. Fantastic. So I think with that, we'll jump right in, and I think I'd like to start in the targeted therapy realm. A lot of work has been done in the past decade and a half or so in researching and discovering biomarkers and appropriate therapies that directly target them. Can we talk a little bit about some of the new findings in the area of targeted therapies? Specifically, let's start with EGFR. There's been a number of studies looking at treatment options for patients that have had progression while taking Tagrisso, and I'm going to use Tagrisso because that's what most patients use when they know what kind of treatment that they're on, which is currently the standard of care for patients newly diagnosed with the EGFR mutation. And maybe we could start with the Chrysalis trial and what was coming out of that.
Sure. So, right. So Tagrisso is, as you mentioned, is the standard of care now for patients diagnosed with advanced EGFR mutant lung cancer. And what I think in the last couple of years, since Tagrisso has been used as initial line of therapy, we also recognize that when resistance happens, there are multiple different ways in which it can happen. And sort of the approaches are to go after those specific ways if you can understand how it happens, or to develop sort of more general mechanism agnostic types of strategies to overcome resistance. So the Chrysalis study is kind of a more of a targeted approach towards resistance. It really combines two different types of medicines. One, an EGFR antibody that sort of binds the outside portion of this receptor, and Tagrisso binds the internal portion. And then using a second compound called lazertinib, which is a kind of a Tagrisso-like medication. And what the study showed at ASCO was that in patients that had developed resistance to Tagrisso, there was some activity of this combination. There was a 30 some plus response rate of patients whose tumor shrank. But it was also clear that it wasn't uniformly distributed, meaning that patients whose cancers had the EGFR protein and the MET protein, which is the other half of this what this antibody binds to, seem to do better. So I think we're, again, hopefully we'll be fine-tuning how we approach resistance, and this combination being studied in this trial, I think, is one example of that.
So before we jump to the next, I want to talk about resistance just a little bit more because it's relevant sort of throughout this
Sure.
area that we're talking about, and just can we give patients a little more information on what that means? I know we get calls all the time with patients fearful of resistance and when is it going to happen and what is that going to look like and what's my plan B? Can you just dig a little bit deeper into?
Absolutely. So I think one thing that we lack information on today is being able to predict sort of when that's going to happen. We can look at things like medians or what's the median time until which that happens. But of course, for any one individual, that person, for him and her, it's a specific point in time. And I think we don't have a great understanding of what dictates if that's going to happen earlier rather than later in the course of treatment. Typically, for things like Tagrisso resistance, it happens relatively gradually. It isn't typically that from one scan to the next, things are sort of completely different. There's usually a gradual process. You start to see some subtle changes, and sometimes in many places, sometimes just in one place. And resistance can be sort of can be in one area or it can be multiple areas simultaneously. The important piece there is to understand why that's happening. And usually that means resampling the cancer somehow, either trying to biopsy the cancer or do blood-based testing, something that will give us information as to which of the many different ways is this cancer now trying to get around the effectiveness of Tagrisso. And if we can understand that, that can help us direct towards specific clinical trials like the Chrysalis trial, which goes after sort of the mechanisms that impact things like new EGFR mutations or MET amplification or other sort of defined mechanisms of resistance.
So I think you brought up a really important point about this sort of reevaluation and or biopsying at progression. I think that's their sort of gray area for a lot of patients and why that is important. And it's not just important with an EGFR patient, right? It's important in general when a patient who's on a targeted therapy progresses.
Yes,
Is that true?
absolutely. Think of it as an evolutionary process. The cancer is trying to evolve to grow around, be it Tagrisso, be it alectinib, be it whatever a targeted therapy. And there's more than one way to do that. And by understanding how, in the specific instance that this is happening, we can better tailor sort of the next therapy for the individual. So absolutely, it's important. It's not just important after the first therapy; it's important after the second therapy. I think the more informed we are about what's happening in the cancer, the better we can sort of tailor and match the therapies to individuals' cancer. So it's a continuous process. You can't rely on the old biopsy because it's changed after the treatment, and so you have to look at a new one. And so absolutely important to continue to do those types of analyses.
And does that rebiopsy have to be on tissue, or can it be on a liquid or blood-based biopsy?
Yeah, it doesn't always have to be. Now, there are some pros and cons about doing tissue-based biopsies versus liquid biopsies. Now, tissue-based biopsies, of course, things have to be in a place where it's safe to do the biopsy. And there's certain parts, for example, something that would be growing next to the heart, where you would not necessarily want to try to biopsy it with a needle because you wouldn't want to puncture a great vessel or anything like that. But the tissue allows the pathologist to look at it under the microscope as well and do sort of protein-based studies. And sometimes we see what are called state transformations, where an EGFR mutant cancer transforms into small cell or has squamous cell features. And those you can capture under pathology. On the other hand, liquid biopsies, you can take blood from everybody. And so there's potentially a greater applicability to be able to study the genetic material that cancers spill out into the blood from a greater number of individuals. The downside is you can't look at it under the microscope, and some of the things that are not necessarily due to a genetic alteration you can't capture. But so there are pros and cons of both. We try to sometimes do both, in fact, because they give you complementary pieces of information when that's possible.
That's great. And I know I don't want to derail us too much off of, the conversation around ASCO and discovery, but you did talk about small cell transformation, and I know we do get a lot of calls about that. Can you explain that a little bit and talk about how common it may or may not be?
Yeah. So what happens is that for reasons that some of which is understood, some of it which is not, the typical, for example, EGFR mutant cancer, which are adenocarcinomas, and treated with Tagrisso or other EGFR inhibitors, about 5 to 10% of the time, they can take this transition and all of a sudden now start to look like small cell lung cancer under the microscope. And they share some of the sort of the genetic features of the more common forms of small cell, and they tend to be treated like small cell lung cancer, which is typically with chemotherapy directed at small cell lung cancer. So it does happen. It's not that common, but it has a very distinct sort of treatment path once we identify it.
That's great. Thank you. And I think we could probably have a whole living room or conversation about that another time. Jumping forward, I want to talk about the HER3-DXd. What can you tell us about that?
Right. So this now falls under, I think, a category that is agnostic to the resistance mechanism. Right. As I mentioned, you have many different ways in which cancers can develop resistance to medicines like Tagrisso. And this is a strategy where that is less important. And what this is trying to take advantage of is this HER3 protein. It's sort of a family member of EGFR. And it turns out that essentially all EGFR mutant cancers also express this family member. It sort of comes along for the ride. And but it's not a resistance mechanism to Tagrisso or any other EGFR inhibitors. It just happens to be there. And what the HER3-DXd is an antibody drug conjugate. So it's an antibody that's linked to a conjugate. And so what happens is that, and the conjugate is kind of a chemotherapy. And so what happens is that the antibody binds the HER3 that's on the EGFR mutant cancers, and then the whole thing is sort of taken into the cancer cells, and the toxin, the chemotherapy part, is sort of specifically released in the cancer cells. So it's kind of like a Trojan horse. You're delivering the chemotherapy, but now specifically to the cancer cells and specifically to the EGFR mutant cells in this case. And in that study, it was found that, again, there's activity of this. It's definitely working. And it's working, again, as I mentioned, sort of the hypothesis was that it didn't matter if you had cancer where you actually knew what the resistance mechanism was, or if you had a cancer where you didn't know the resistance mechanism. And in fact, it works just the same in both. So it's a totally different strategy, but it's a new option, and there are additional clinical trials ongoing with that as well.
Thank you. I want to pause just for a second to remind folks who are watching, because this is complicated stuff, right? If you have any questions, please don't hesitate to type them into the live stream so we can get those answered for you. One other area where there was some recent exciting announcements, I think back in May, is in the rare sort of EGFR exon 20 mutation. What's going on in that space?
Yeah, so there is the EGFR mutations kind of fall into sort of three buckets. There's the common ones, the what we call the exon 19 and L858R mutations, where we just talked about Tagrisso, which are about 85% of EGFR mutations. And then there's sort of two rare categories. One, which we call uncommon EGFR mutations, so sometimes these G719 mutations and others. And then these exon 20 insertions. And each of these two other categories make up about 5 to 10%. The exon 20 insertions are just like any other EGFR mutation, except because of where they happen to occur in EGFR, we as a scientific and medical community have had a tough time finding medicines for these and developing specific drugs. And so in May, we had the first ever approval of a drug for the exon 20 insertion, a medicine called amivantamab, which is we talked about this Chrysalis trial. It's the antibody in that trial, but here used as a single drug, EGFR MET dual specific antibody, and definitely had some activity and enough that led to a regulatory approval specifically in this exon 20 space. So exciting time because we've not had a therapy in this space, and despite trying for many, many years. There are a number of other medicines, sort of Tagrisso-like medicines, which are pill medications, whereas the antibody is an IV medicine, that are being tested in this space as well. So we anticipate to see more activity in this space moving forward. There was one presented or a couple presented at ASCO this year. One called DZD9008. So when they're early drugs in development, they just have letters and numbers. And then this medicine from Cullen and Oncology. And both definitely had some activity, and I think both of them look to be very good in terms of moving forward and with additional clinical trials. So we anticipate that in this space, in the exon 20 space, which is again a rare subtype and a very heterogeneous subtype of EGFR mutation, that we'll continue to see more trials, hopefully more approvals, and we're excited that we had one in May.
That's great. So I want to jump into KRAS now, because I know for a very, very, very long time, there was not a ton of options available for this subtype. So let's start with the CodeBreak 100 trial.
Yeah, so just to echo that and give some context, that KRAS mutant lung cancer makes up a fairly large percentage of lung adenocarcinoma, and about half of that is the KRAS G12C mutation. So KRAS G12C mutation, which is the common type of KRAS mutation, accounts for probably roughly the same number of patients as EGFR mutant lung cancer, at least in the United States. This is where we now have this first approval of this medicine called sotorasib, a KRAS G12C inhibitor made by Amgen, where again the trial at ASCO showed kind of longer follow-up, again, confirmed the responses. It's about a high 30% response rate, and looked at some of the other sort of biomarkers or concomitant biomarkers associated with that, and kind of show that it works across sort of the different concomitant genetic alterations that also happen with KRAS G12C. There's a lot of activity in the KRAS space. And we went through, 10 years ago, or a little bit longer, there was efforts to study KRAS and use therapies against KRAS, and they all pretty much failed. And there's been a complete resurgence of therapies for this, with now the first being these G12C inhibitors. This one from Amgen, there's another one from Mirati called adagrasib, and multiple other ones in clinical development and preclinical development, including multiple novel combinations. So KRAS is the big next frontier in terms of therapies and therapeutic approvals. We're excited about the first one. We anticipate hopefully that the adagrasib one will also get approved. But the success has catalyzed the efforts and interest to go back and say, hey, now with knowledge that we have today that we didn't have 10 or 15 years ago, we can do better. We understand the biology better. We understand drug development better. We can develop therapies, and that will continue to grow. And that's a really exciting development because it's such a big population of our patients.
Sorry, that's great, Pasi. And I don't know, I'm getting a weird sound. I just want to make sure that that's just on my end and not everyone else's before I move forward.
A little ticking.
Does everybody else sound fine?
Talking
Yeah, it's okay.
Okay.
Yeah.
Okay. I missed a large portion of what you said, so I don't know unless there's another question that came out of it. It was probably just me, so I can't pick up where you left off to kind of forward think about what a patient might ask. So go ahead, Bonnie.
I have a question. And how often does a new mutation pop up in a lung cancer patient upon progression, where maybe it wasn't there at the beginning, or it was, but then they started progression, and oh my goodness, here's something new.
Yeah, fairly often. I would say that just like genetic alterations give rise to sensitivity, the vast majority of resistance are through genetic alterations. So they're mutations or amplifications or others. So they're fairly common, but still there is a substantial portion where we don't have anything. But of the common ones, it's usually a mutation. Now, we don't know that we can necessarily always target those or have a drug against a specific mutation, but genetic alterations drive resistance fairly commonly.
Great. Okay, I'm trying to be mindful of time because I know we have a ton of really amazing stuff to go through. Let's jump to HER2. What's going on there?
So HER2 mutant lung cancer is a subset of lung cancer, smaller than EGFR mutant lung cancer, makes up about 2 to 4% of lung cancer, and something that has therapies that that are often used in breast cancer, where HER2 amplification is common, have also started to migrate into the HER2 mutant space. We don't have an approved therapy for HER2 mutant lung cancer. Probably the most exciting data for HER2 mutant lung cancer is using a medicine called trastuzumab deruxtecan, or sometimes also known as Enhertu, which is again another one of those antibody drug conjugates, those Trojan horses that I mentioned earlier. This is a drug that's approved in breast cancer as well as in gastric cancer. And at ASCO in 2020, there was exciting data on it in HER2 mutant lung cancer, which will get updated later on this year. And the data so far looks quite good, with over half the patients having responses. It's much better than has been seen with the kind of a small molecule pill medicines that we use for HER2 mutant lung cancer. And we anticipate, or at least we hope, that this will be the next subset of lung cancer where we'll have an approved therapy. But so far, it looks exciting. There are still other HER2 small molecule inhibitors out in clinical development, and so look out for those as well. But I would say the frontline leader in the sort of the targeted therapy space here is the trastuzumab deruxtecan.
How often is HER2 found in lung cancer?
So mutations are found in about 5%, 3 to 5% of individuals. You can sometimes have HER2 amplification in lung cancer like you do in breast cancer, but it's exceedingly rare. It's usually 1%. Sometimes it can be a resistance mechanism to things like EGFR inhibitors. HER2 immunohistochemistry. So now we're not talking about the genetic alteration, but the protein that can maybe be found up to a quarter of lung cancer patients. And in fact, this trastuzumab deruxtecan drug was tested in lung cancer patients, not based on the genetic alteration HER2, but just the presence of the HER2 protein. And it had some efficacy. It just wasn't quite as good as it is in the HER2 mutant population. So whether it'll find a home in the protein expression patients is unclear at the moment. But for the mutant patients, absolutely.
So I think I heard you say that a breast cancer drug is being repurposed in this space. How often is that happening? I mean, we're in this new era, right, of all of these biomarkers sort of driven cancers. Are we seeing more of that with this repurposing of drugs that were typically used for another type of cancer?
Yes, and I think one thing that we don't always know is that is a mutation in one cancer the same as a mutation in the other cancer, meaning that if you have the very same mutation in a breast cancer versus lung cancer, do they behave the same or not? The other example that in lung cancer is the BRAF mutation, the BRAF V600E mutation, found very common in melanoma. And the same combinations of medicines that are approved in melanoma work in the lung cancer patients that have that same mutation. But if you have a colon cancer and have that mutation, these medicines don't work. And so the context in which these mutations exist is important. And getting back to the KRAS example, so KRAS G12C mutations with the KRAS inhibitors work in lung cancer. And as you mentioned, this is now an approved therapy. The sotorasib is an approved therapy. However, in colon cancers with the same mutation, these medicines work much less. And part of what we as a community are trying to understand, and what's so important about doing clinical trials is to try to understand is the medicine against the same mutation work similarly or differently in a different disease type, and by understanding why they may work differently may also help you think about what are the appropriate things that you can combine with that medicine to make it work better. That may impact both lung cancer and colon cancer in this situation.
Yeah.
Interesting.
Is that similar to having an exon 20 with EGFR versus no exon 20? Does that make any sense?
Although it would be although thinking about the EGFR exon 20 in another cancer, and there are very few other cancers that have EGFR exon 20 mutations, but there are HER2 mutations in other cancers. So, for example, even breast cancers, some breast cancers have HER2 mutations. We also find them in bladder cancers and in other cancers. And as we as a community do more systematic sort of biomarker testing just across different cancers, you occasionally find mutations where you didn't expect, and that opens up a therapeutic opportunity for that individual very much. So I think that that's an important thing as well.
Exactly, because when we talk about each patient being very, very unique and different than another patient, could it be those mutations that are below the major, like exon 20 and 19, etc.? That's where we're going to find why one therapy works and where one doesn't.
Yeah, I think the context in which these mutations live in, even when we talk about things like EGFR mutant lung cancer or KRAS mutant lung cancer, those aren't the only alterations in that cancer. They may be the dominant alteration, but there are others as well, and those others may impact on the durability of a treatment. And it's just something we're just, I think, at the infancy of trying to understand. We've been so focused on finding the right alteration, matching the drug. I think we're now getting to sort of a next level of precision medicine where we're saying, okay, what else is there, and how else do these things impact things, and in a positive way or in a negative way? And if we understood that, it may help tell us and inform us about the treatments and whether they should be a single drug or a combination, depending on what that is. So I think we're becoming more refined in thinking about the precision medicine, and that will be part of the next phase for the future.
Exciting. Very exciting.
Yeah,
great stuff. So I want to jump to Rhett really quick because I believe some longer term updates came out of ASCO this year.
Yeah, so right, so we have two approved RET inhibitors, pralsetinib and selpercatinib, both of them which are highly effective in RET rearranged lung cancers, and efficacy can last for a really long time. So really a welcome addition to our armamentarium of targeted therapies for lung cancer. And RET alterations are now things that we routinely test for in genetic testing to find individuals so we can get them onto these effective therapies.
That's great. And I think, in addition to a lot of, the targets that we've mentioned earlier, I think there were several early stage trials that were looking at targets that really, prior to this ASCO, I wasn't aware of. And I think it's just worth mentioning. It's PARP7, NRG1, SHP2, and GLS1. I don't know if there's anything really to report on that, because I know they were really early stage trials. If there's anything you want to say to that before I kind of jump into my...
Yeah, these are early stage trials, and but important biology wise. So, for example, SHP2 or SHP2 is an important component in the sort of the signaling in KRAS mutant cancers, for example. So there are several medicines and several companies developing SHP2 inhibitors, either as single agents or combined with KRAS inhibitors to sort of perhaps augment the effect of KRAS inhibition. And many of the trials are still kind of in the early phases, making sure can you combine these two medicines? What's the right sort of dosing and schedule of being able to give these medicines together? But I think all of these targets that you mentioned are good ones, and I think will again add to the list of agents that we all will be able to use either as single agents or in combination.
Yeah, and before we jump into the immunotherapy section of this, I kind of want to do a little rant about biomarker testing, because there was some information that came out at ASCO. It was data from a My Lung Consortium and the Flatiron Health Records data that demonstrated that some significant testing gaps are still there. They still exist, particularly in Black lung cancer patients. They're receiving biomarker testing at lower rates than white patients, despite having the same rate of targetable mutations. And also that while the majority of patients in the US are receiving some biomarker testing, most are not being tested for every biomarker with an approved targeted therapy, let alone a full comprehensive panel that may qualify them for one of the trials that we're talking about today. So I say this because this data is extremely relevant to GO2 Foundation's one of our strategic priorities of ensuring comprehensive testing for all patients diagnosed with lung cancer. So I don't know if you want to rant along with me and say a little bit about the importance of biomarker testing.
Absolutely. I think, this is, we recognize that lung cancer is a heterogeneous disease, and in order for us to be able to match the best therapies for the specific cancer, you need to do comprehensive testing. We have multiple drugs approved for specific genetic subtypes. You won't find them unless you do comprehensive testing, and you can't guess. You have to do the testing. It's absolutely critical. It's a standard of care in 2021 for every lung cancer patient to get tested. And I think if you as a patient haven't been tested, please ask your provider to do so. It's absolutely standard of care.
Yeah, yep. Amen. Singing our song. So I want to jump into immunotherapy, particularly for metastatic non-small cell lung cancer. What can you tell us about what's going on in that space?
Yeah, I think there are sort of multiple, and kind of what was presented at ASCO was some of the long-term follow-ups there, and there are, I think, choices when somebody when immunotherapy is the first line therapeutic choice for somebody. There are sort of multiple choices in terms of how to best deliver that. Meaning, should it be immunotherapy alone? Should it be immunotherapy with chemotherapy? Should it be combinations of immunotherapy, or should it be sort of the kitchen sink approach of combinations of immunotherapy with combinations of chemotherapy? And there are different instances in which one may use one versus the other. Many of these different approaches haven't been totally compared head to head, so it's sometimes a little bit hard to know exactly which is the right approach to use in that specific situation. There are pros and cons of whether one would give chemotherapy with immunotherapy or immunotherapy by itself, and so a lot of movement in that space, and especially in the sort of frontline space of systemic therapy, and trying to understand sort of the biomarkers of immunotherapy for the targeted therapy space. The biomarkers are, it's either there or it isn't. It's a very sort of dichotomous thing, and for immunotherapy, it's much less clear. It's sort of a degree of PD-L1 expression or other biomarkers. It's not so categorical, and I think it's been more of a challenge there. But the good thing is we do have a lot of immunotherapies approved, and I think those that provides great options for our patients.
Yeah, I think this is an important sort of topic, even outside of what's currently happening or what new, we may be looking at, because, the pharmaceutical industry has done a great job of this direct to consuming, right? So I have to tell you, and I know the calls come in all the time about, well, I want immunotherapy because they said X, Y, or Z. How do you determine, how do you answer those questions for your patients, right? Because I'm going to stop there and just ask how you deal with that when you get those types of.
Yeah, I think we have to recognize, just like targeted therapy doesn't work in everybody, immunotherapy doesn't work in everybody, and we also recognize that for a lot of the patients that have the targetable alterations, like the EGFR and ALK and ROS and RET alterations, et cetera, these are populations in which immunotherapies tend not to work very well. The exceptions on the targeted space are KRAS mutant cancers and BRAF mutant cancers, where you do have some effects of immunotherapy. And so first, I think it's, we sort of approach the idea of, is there a targetable alteration in someone's cancer? And if so, we treat them with a targeted therapy. If not, then I think immunotherapy comes into play. And oftentimes the PD-L1 expression in the cancer is an important determinant. If it's very high, often can think about using single agent immunotherapies, or whereas if it's not, we tend to reflex back into chemotherapy and immunotherapy. So I think it's trying to understand, again, it's putting it into the therapeutic context of where does that fit in, and where does it fit in relationship to what else is happening in that individual's cancer.
Perfect. Okay, I want to jump to early stage non-small cell lung cancer, because this has been a hot topic, I know, since ASCO last year. So I want to kind of start there before we jump into what's sort of grown from that, particularly as it pertains to ADAURA, and what research is continuing to look at in the not only the adjuvant space, so the post-surgical space, but the neoadjuvant or the pre-surgical space.
So I think this is a fantastic evolution in targeted therapy and in immunotherapy for lung cancer. All of the things that we just talked about, are really for our patients with advanced cancer, and that's where a lot of the advances have been made. And we're now finally, over the last couple of years, starting to see these things migrate to being used earlier and earlier, because we recognize that even with the most successful surgical approaches, patients still do relapse from their lung cancer. And at the moment, the approved therapies after surgery are chemotherapy in the adjuvant setting, which does help, although the help is the effectiveness is perhaps more marginal. And so, since we're matching our most effective therapies to patients in the advanced disease, why don't we use it in the earlier disease as well? And the ADAURA trial that you mentioned from last year looked at giving adjuvant Tagrisso specifically in EGFR mutant cancers, in which patients had undergone surgery, and had just a dramatic reduction in the rate of relapse of individuals compared to not giving an EGFR inhibitor, which led to the regulatory approval of Tagrisso in that setting. Now, what we don't know, and what's true for many of these trials, is of course that our goal in the early stage setting is of course to cure more people. And what we don't know from the ADAURA trial, for example, is that does the addition or does the use of a medicine like Tagrisso after surgery actually do that or not, or does it just delay the likelihood that if someone's cancer is going to come back, it's going to come back? And part of the reason is that because that endpoint takes so long to happen, but the data was so impressive that it led to regulatory approval. This year at ASCO, we saw the first example of giving now an immunotherapy in the adjuvant setting. So post-surgery, post-chemotherapy, immunotherapy versus placebo. And similar to what we saw with ADAURA, there was an improvement or a reduction in recurrence if you got immunotherapy. Again, too early to say, does that lead to more people being cured from their lung cancer or not? And still the follow-up in that trial was earlier, and we need to understand more of the biomarker subsets there. And is it in everybody who's had surgery, is immunotherapy effective? Or is it just like we think about it in the advanced disease, that cancers that have this high PD-L1 of greater than 50%, is that the population where this is going to be effective? So more information to come there, but certainly, again, an exciting piece of information there. And many, many other trials are underway in this adjuvant setting. So post-surgery, post-chemotherapy, getting a targeted therapy. And again, more to come over the next several years. Now, neoadjuvant is giving it before surgery. So, and at AACR this year, we saw an example of a very exciting a study where individuals were given either chemotherapy before surgery or chemotherapy and immunotherapy before surgery. And essentially, there was a tenfold improvement in the sort of what we call the complete pathologic response when you got chemotherapy and immunotherapy, meaning that at the time of surgery, when the pathologist went and looked at it under the microscope, there was no viable tumor there. So our hope, again, there is, and it's too early to know, is that if the effectiveness of the therapy has been so good in the beginning, and now anything that's left there has been removed, that means that will improve or translate into a reduction in recurrence. Time will tell, but we don't know that yet. There are a number of these trials underway with immunotherapy, and again, also in the targeted therapy space. If you find an EGFR mutation prior to surgery, giving individuals Tagrisso or chemotherapy Tagrisso. Again, the idea: at surgery, have you seen this dramatic effect at surgery, and then ultimately will it translate into a reduction in recurrence, and ultimately, of course, an improvement in cure? So I think it's an exciting time, because removing the effective therapies that we've worked so hard in the advanced setting to now also incorporate into the earlier setting. And it's also also an important piece, because it means that we all have to work together as a community, our surgeons, our radiation oncologists, our medical oncologists. It takes a village, right? And I think everyone, now that we're having effective systemic therapies, of course, and local therapies, I think we'll hopefully, again, continue to see the needle move and have more people cured from their cancer.
So, Dr.
Yeah,
Jänne,
I love that.
Just about circulating tumor cells and being proactive for the early stage patients by giving them chemotherapy plus radiation, maybe immunotherapy, etc., and then the Tagrisso. In my mind, it seems to me that if we catch those floaters, in the body, so to speak, early, does that give the patient a better chance to survive a much longer time?
Well, I think we're seeing fantastic technological advances in being able to detect this circulating DNA that's tumor derived, and we can now do it even in patients who've undergone surgery. So the technologies are better, that they can detect smaller and smaller quantities. And studies to date have certainly shown that if after surgery you still have detectable DNA in the blood, the likelihood of recurrence is much higher than if you don't have detectable DNA in the blood. And what we haven't done yet is use that as a way, as you point out, to say, okay, should we direct treatment based on that? In the adjuvant setting, right, when I mentioned that we give people chemotherapy and chemotherapy has an effect, we're probably over-treating a number of individuals. There are people who are probably just cured from their surgery and don't need the chemotherapy, and hence won't never benefit from the chemotherapy, may just get side effects. If we had a way to identify those individuals who are in need of the adjuvant therapy, be it chemotherapy, immunotherapy, Tagrisso, whatever, again, we can tailor our therapies to those individuals that need it. And I think this will be a technologic advance that we will now start to incorporate into trials, and are already doing that, to be able to, again, tailor our therapies to those who really need it. So I think it's an important evolution, and it's an important area, and we're getting there.
Yeah, I think it's such an important conversation having right now in this early stage arena, particularly with GoTo's efforts and others trying to get more and more folks into screening so we can detect these cancers early. But what we do know, and correct me if I'm not remembering my numbers correctly, but roughly 40%, give or take, of folks who go on to surgery will have a recurrence. So knowing that, the important work that's needing to get done on top of, we're trying to get people screened and get them in the door so they can, be diagnosed early, only for them to say, oh, well, I relapsed and you didn't have anything. But now here we are, I guess, is my point.
Yeah, I think our tools are better in that. and we want to obviously find people earlier, so surgery is going to be more successful the earlier stage the disease is, before it spread to lymph nodes, things like that. So, absolutely, we're getting there, which is good. There's a lot of excitement in this space.
Yeah, that's great. So a question did come in from online, and I think it's an appropriate question for any time during this conversation. But when offered a clinical trial, when should a patient consider it a valid option? Are clinical trials typically successful or more high risk / reward?
Well, I would say there are different types of clinical trials. And I think for example, we try to have clinical trials for essentially every treatment decision, be it initial diagnosis when we're thinking about a therapy, or after their initial therapy has stopped working, or et cetera. So there are various stages in which one can participate in a clinical trial. And clinical trials are how we get new therapies to the market. And, we need to, in order for us to know, is a therapy working, we need to do a clinical trial. We need to test it. And sometimes those are from early phase trials, whereby they're just sort of entering the clinical space where we know less about them, or they're more advanced, or sometimes they're what we call phase 3 trials, where it's sort of the final step before approval of a drug. But it's an important piece, and I think participation in a clinical trial is a way in which we move the field forward, and I think an important thing. We don't. If we knew the result of the trial, we wouldn't do the trial. So, getting to the question of are they going to be successful, we don't always know the successes. We sometimes have better ideas of whether or not they're going to be successful, depending on where they are in the sort of the life of that trial. But there's usually a question to be asked in the trial that we don't have an answer to, and that's the reason to do the trial.
So is it true, because I think a lot of folks are fearful of even talking to their physicians about clinical trials, because they're thinking of their grandfather or great-grandfather's clinical trial where they're getting a placebo or a sugar pill versus a study drug. In most cases, is it not true that you're either getting standard of care or study drug?
Yeah, that is correct. I think there are very few trials at the moment that basically compare an active therapy to no active therapy, a complete placebo. You may be looking at a new therapy by itself. You may be looking at a new therapy in compared to an existing therapy that's an approved therapy. So I think it's always worth a conversation though with your provider and asking those details. But most of the trials that we do these days don't involve placebos, i.e., where there's no therapy. There's usually some therapy or comparison of two therapies, or just a new therapy being evaluated by itself. But definitely talk to your docs about that.
I think that's great. I think it's so important. I think it's a perfect opportunity for GoTo and the lung cancer community in general to just thank folks who do participate, because without you, these simply couldn't happen. And I think they're, in my opinion, not a medical opinion, just my opinion, they're worth having a conversation with your doctor about, because it may be right for you at some point during your treatment experience. So I want to jump from that to small cell lung cancer. It's such an important conversation. I know at GoTo, another one of our sort of strategic focuses is to help generate a community around this subset of lung cancer patients. And so before we jump into some early data that was coming out of ASCO, can you talk a little bit about some fairly recent approvals in small cell and why they're so important given the amount of time, since the last approval there?
Yes, small cells, I think historically have been been a little bit more challenging disease to develop new therapies for. Unlike in the non-small cell, where we just talked about all the targeted therapies and genetic alterations, we don't find them in small cell, and that has limited some of these therapeutic approaches. Last couple of years, we've seen a few approvals of new drugs in small cell, which is great. So immunotherapy is one approval in combination with chemotherapy in small cell. So that has been definitely a welcome improvement, and we're using that at the moment. The second is another medicine called lurbinectedin, which is another chemotherapy medicine used in sort of later lines of small cell. So there is some movement. It continues to be a significant portion or significant minority of lung cancer patients, but an important and large minority, and something where we continue to try to find new therapies. I will say that some of the biology, there's been greater understanding of biology of small cell. We are starting to think about it in different categories now as well. So I think that too will, again, when we link biology to therapy, we typically we tend to have more successes like we have in the non-small cell space.
And I think that's important to kind of talk about a little bit further, or further sort of explain why it's important to know the biology. Because at some point, and it was very similar in non-small cell, right? Until we knew the biology, and we couldn't really figure out what these biomarkers or future biomarkers might look like or be.
Yeah, right. I think different subsets may have different biomarkers, and the different subsets may have different therapeutic vulnerabilities, and knowing those subsets will be important. I think in small cell, one of the challenges that has sort of perhaps hindered some of the development is that we tend to not do surgery in small cell, so you don't have sort of specimens and tumors in which you can do extensive analyses. It tends to be a kind of a very central disease, so often the biopsies are very small, and there isn't an opportunity to do these sort of extensive genetic and other biomarker analyses, which we've been able to do in non-small cell. So some of those challenges, I think, have also limited some of the development opportunities. But I think, again, with newer technologies, we're able to do more with smaller specimens and things like that. So I think that's helped push this forward.
Great. Can you talk a little bit about the early data that was presented on the drug that targets the DLL3?
Yeah, so DLL3 is a unique protein that is found on the surface of small cells, and this has been a therapeutic target for some time. Actually, several years ago, there was a medicine called Rova-T, which was a antibody drug conjugate, again, one of these Trojan horse medicines that specifically targeted this protein on the surface of small cell lung cancers. Now, there was sort of good news and bad news about that trial. The good news was that there was some efficacy. It showed as a proof of principle that if you target this DLL3 protein, you can have responses. There is a way to turn that into a therapy. The bad news was that the Rova-T medicine turned out to be too toxic, and this conjugate kind of non-specifically was loosened off the antibody, and people ended up having side effects. So companies have now gone back and said, okay, it's a good target. This is what the Rova-T proved, and now let's try using other approaches like this medicine that we saw at ASCO, this sort of bispecific antibody. And so we're starting to see early responses there. Completely different way of inhibiting DLL3. We don't see the sorts of side effects that we saw with the Rova-T medicines. Again, hope remains that this could be the targeted therapy for small cell that we've been waiting for, if we can now target it in a better, safer way, and than we were able to before. So again, it's a great thing. Lots of excitement about that, and hope to see more of that moving forward.
Yeah, I love it too, and I definitely hope to see more of this in the small cell space moving forward. But if typically small cell lung cancer patients are not receiving biomarker testing, how do they know whether or not they have this DLL3 or anything that may come down the pike?
Yeah, I think that is at the moment. I think that's really in the clinical trial space to be even tested for this biomarker, because there are no sort of uniform assays or anything like that. But hopefully, if the trials turn out to be successful and you can show that if your cancer has this DLL3 protein, that this medicine is effective, usually that will then drive the biomarker testing, which we don't, as you point out, we don't currently do in small cell. But if there's a successful therapy, we will start to do it for sure.
Great. Anything else about small cell, or did we kind of cover the meat of what was discussed?
I think we covered the meat. I think that despite it being a challenging area for new drug development and new therapies, I think that there is some hope with some of these new approaches, and we again hope that will translate into effective therapies.
That's great. Again, being mindful of time, we have about 5 minutes left. I'm going to ask both of you if you have any final thoughts before I go through some housekeeping. And Bonnie, I'll start with you.
Okay, this is a great opportunity. I would love to see about 10 people, the village, the people that make a village, sit down and talk about the next 5 years, what they're going to look like, and then make it happen. What do you think, Pasi?
Absolutely. Absolutely.
Yeah.
Absolutely.
Right.
I think I think we look at, despite the last year and a half being a difficult year for everybody with COVID, I think it overshadowed a lot of the excitement we've had in the lung cancer space with the number of new drug approvals in this space over the last year and a half and new indications. And it's a testament to us understanding more of the biology, what's happening in cancers, how are they evolving, and I think I think the next phase is to figure out how do we leverage these successes and continue to move the needle forward, as you point out, and
Right.
let's do it. We have opportunities here. We just need to move them forward.
Exactly. And we can, right? We can do it.
We can.
That's great.
We can.
That's great.
Yeah.
I think that sums it up, and I can't thank you both enough for this conversation tonight. And anybody who's watching online who maybe didn't get a question answered, please reach out, and we will definitely get it answered for you. A huge shout out to Pasi, Dr. Jänne, for coming and talking with us tonight about Best of ASCO. We really hope that eventually when we get back to live and in-person meetings, he will come and join us out in California for a meeting where all of you can be present with us. Although this has been a nice way for us to continue to bring you this information, it's certainly not the same as all being together. So thank you. As always, I'm going to do some of my closing and housekeeping and let you guys know what's coming up next. I'm also going to remind you about our living room community. You may have seen the URL flash along the bottom. So if you want more information about what's going on in the living room, behind the scenes, up front, or otherwise, please go to gotofoundation.org/living room and sign up, join the community. Come be one with us. For those of you who may not have received Bonnie's book yet, you can still find information about where to get that on our website, gotofoundation.org, or you can simply go to Amazon and get your copy there. I'm pretty sure that everybody who watches The Living Room has seen us talk about her book. It's called The Living Room, A Lung Cancer Community of Courage, and it highlights 20 individuals, with varying aspects of disease, varying ages, from all different backgrounds, and really are stories of hope. And you don't have to have lung cancer to really get something out of this book. So I encourage you guys to do that. Our next living room is August 17th. Where we'll be talking about financing your lung cancer care. I know that that's a challenge for a lot of you out there, especially as these drugs are newly approved. Sometimes they aren't always covered. So Clara Lambert from TaylorMed is coming. She's the Director of Financial Navigation. We're having a great conversation about that. Our upcoming events, we are happy to say that our nationwide golf event is going to be on August 20th through the 22nd. Anyone can join from anywhere. Visit golfforgoto.org. And in-person events are back. So registration is now open for our San Francisco and Philadelphia events taking place this fall. You can find more information about that on our website as well. They will be hybrid, so you don't have to come and join us in person in order to participate. So please consider doing that. We talked a lot about research tonight. And so for those of you who were watching, I assume that research and what's coming out of it are important to you. So please consider joining the Lung Cancer Registry at lungcancerregistry.com. I always hate this part. It sounds like such a commercial, but it's where you can donate your information. It's patient-reported outcomes about varying aspects of the disease that we can then share back with the researchers like Dr. Jänne so that maybe they can find some insights. So I welcome you to do that. And our Magnifying Lens, which is a newsletter coming out from our Science and Research Department, might also be of interest to you as well. So please consider some of these things. I'm going to finish again with a huge thank you to Dr. Jänne, all of you watching live or post-live, Peninsula Television, and the GoTo team. Of course, our supporters, without their support, we simply could not bring this to you. Amgen, AstraZeneca, Bristol Myers Squibb, Daiichi Sankyo, Foundation Medicine, Genentech, Lilly Oncology, Merck, Novartis, Regeneron, Sanofi, and Takeda. Thank you from the bottom of our hearts and for believing in us and this program. Some of you may have seen some link come across the screen, and I promise this is the last thing I'm going to say, for donations. This by no means, it means it's a requirement for this program. The Living Room will, of course, always remain free programming to anyone and everyone who can benefit from it. But we thought a gentle reminder for those of you that could or felt so inclined that we are a nonprofit, and along with all of our amazing supporters that we just announced, we do rely on the generosity of our community to do all that we do for you. So with that, I'm going to close out. It is 6:30 on the dot. Sorry I spoke so quickly at the end. Hope to see you all next time in August. Until then, enjoy your summer and be safe wherever you are. Good night, everyone.
Oh, I'll be brave if you're brave. I'll be brave, but only if you're brave. And it could be just you and me. We'll be family. Just wait and see.